Welcome to this lecture on the fundamentals of biomolecules. Today, we are going to explore the fascinating microscopic world of simple sugars, specifically focusing on aldoses and ketoses. You might not realize it, but the exact placement of a single chemical group in a sugar molecule dictates how your body harvests energy, digests food, and builds complex cellular structures. By the end of this video, you will understand exactly how these tiny structural differences create massive biological impacts. Now that we know what we are studying, let us start with a simple analogy. Imagine a long freight train carrying cargo. The way that train operates depends entirely on where the engine is placed. In biochemistry, simple sugars, or monosaccharides, are like these trains. They are essentially chains of carbon atoms, and their reactive engine is a chemical cluster called a carbonyl group. If nature places this reactive engine at the absolute front of the carbon chain, we call that sugar an aldose. However, if the engine is tucked away somewhere in the middle of the chain, we call it a ketose. This simple placement is the master key to their behavior. Building on that train analogy, let us look at the formal scientific definitions. An aldose contains an aldehyde group, meaning the carbonyl carbon, represented as C double bond O, is located at the absolute end of the chain and bonds to at least one hydrogen atom. On the other hand, a ketose contains a ketone group, where the carbonyl carbon is situated internally and bonds to two other carbon atoms. In both types, all other carbons attach to hydroxyl groups, written as OH. Because they share the exact same building blocks but arrange them differently, aldoses and ketoses of the same size are known as structural isomers. They have the same chemical formula, but vastly different chemical behaviors. Understanding this structural difference leads us to ask why nature bothers creating two versions. The placement of that carbonyl group drastically alters how the sugar interacts with enzymes in your body, much like a differently shaped key fitting into a different lock. For example, glucose is an aldose and serves as the primary immediate energy source for your brain and muscles. Fructose, however, is a ketose found in fruits, and your liver must process it differently before extracting energy. Furthermore, the aldehyde group in aldoses makes them highly reactive, reducing sugars. This allows them to link with proteins for cellular communication, though unregulated linking can cause damage in diseases like diabetes. To truly understand these sugars beyond their biology, we must look at how we represent them mathematically. All simple monosaccharides follow a basic stoichiometric formula that predicts their exact composition. The formula is CnH2ON. Here, the variable N represents the total number of carbon atoms present in the sugar molecule. For every one carbon atom, there is exactly one water molecule equivalent, which is why they are called carbohydrates, meaning hydrated carbons. If the value of N increases, the sugar molecule becomes longer and heavier. This physical change directly alters properties like how easily the sugar dissolves in water and how sweet it tastes to us. Now that we have our empirical formula, let us see this with a concrete example. Suppose we are analyzing a sugar with a carbon backbone of 6 atoms. We are given that N equals 6, and we need to find the exact chemical formula. We start by substituting 6 into our general formula, CnH2ON. By distributing the 6 to the water molecule equivalent, we get 6 carbons, 12 hydrogens, and 6 oxygens. The final answer is C6H12O6. Physically, this exact chemical formula applies to both glucose, which is an aldose, and fructose, which is a ketose. proving they are indeed structural isomers. This formula we just used tells us the atoms, but it brings us to an important question about their three-dimensional shape. Because the carbon atoms in these sugars are attached to 4 different chemical groups, they act as chiral centers. A chiral center creates a specific three-dimensional geometry that cannot be superimposed on its mirror image, much like your left and right hands. This spatial arrangement is absolutely crucial because enzymes in our bodies are extremely picky. They will usually only recognize and bind to one specific three-dimensional shape out of all the possible variations. Therefore, counting these chiral centers is essential for understanding biological activity. Knowing that chiral centers dictate shape, we can calculate the total number of possible stereoisomers using a simple mathematical rule known as the Lebel-Van Hoff rule. The formula is n equals 2 to the power of c. In this equation, the symbol n represents the total maximum number of possible stereoisomers, or three-dimensional shapes, for the molecule. The variable c represents the total number of chiral carbon atoms present in that specific sugar chain. As the number of chiral centers, c, increases, the number of possible shapes, n, grows exponentially. This means even a small sugar can have many different spatial arrangements, though usually only one is biologically active. Let us apply this exponential formula to analyze D-glucose. We are given that glucose is an aldohexose, meaning it has 6 carbons, and we need to find its total possible stereoisomers. Because the carbonyl group is at the very top, at carbon 1, it turns out there are exactly 4 chiral centers, so C equals 4. We choose the Lebel-Van Hoff rule, n equals 2 to the power of C, to find the variations. Substituting 4 for C, we calculate 2 to the 4th power. The final answer is 16. Physically, this means there are 16 different possible three-dimensional arrangements for an aldohexose, and D-glucose is just one specific active shape out of those 16. Now that we have used this formula and found 16 isomers, let us understand where that C equals 4 comes from conceptually. We're setting out to prove why an aldohexose has exactly 4 chiral centers. When we examine the 6 carbons in the glucose chain, we look at their bonds. Carbon 1 is part of the double bond to oxygen, meaning it only connects to 3 things, not 4. Therefore, it cannot be chiral. Down at the bottom, carbon 6 is attached to 2 identical hydrogen atoms, so it also fails the chiral test. This logical elimination leaves only the internal carbons, 2, 3, 4, and 5, as true chiral centers, proving our value of C equals 4. Before we move on, let us see this with another concrete example by analyzing D-fructose. We know fructose has the same 6 carbons, but it is a ketohexose. We need to find its total stereoisomers to compare with glucose. Because the ketone group is tucked inside at carbon 2, the number of chiral centers drops to 3, so C equals 3. We again use the formula n equals 2 to the power of C. Substituting 3 for C, we calculate 2 to the 3rd power. The final answer is 8. Physically, this tells us that simply moving the carbonyl engine from the end to the middle cuts the number of possible three-dimensional arrangements in half, from 16 down to 8. So, we have proven that glucose has 4 chiral centers. This brings us to proving why fructose only has 3. We're establishing the mathematical reasoning behind the drop in stereoisomers. In the fructose chain, the carbonyl group is at carbon 2. Because carbon 2 is double bonded to oxygen, it cannot be chiral. Furthermore, both carbon 1 at the top and carbon 6 at the bottom are attached to two identical hydrogen atoms, eliminating them as well. By removing carbons 1, 2, and 6, we are left with only carbons 3, 4, and 5 bonded to four different groups. This proves why C equals 3 and shows the profound impact of the carbonyl group's position. Now that we have calculated and proven the properties of these sugars, let us look at them visually. On the screen, you are seeing Fisher projections, which are two-dimensional representations of these three-dimensional molecules. On the left is D-glucose. Notice the red highlighted region at the very top at carbon 1. That is the aldehyde group, making it an aldose. On the right is D-fructose. Look at the blue highlighted region at carbon 2. That is the internal ketone group, making it a ketose. You can clearly see how carbon 1 in fructose is just a standard CH2OH group, unlike glucose. This diagram perfectly illustrates how a tiny shift in atomic arrangement completely changes the molecule's identity. We have covered a lot of ground today, so let us summarize the most important ideas. First, the fundamental difference between an aldose and a ketose is simply the placement of the carbonyl group, at the end for aldoses and internal for ketoses. Second, this seemingly tiny shift dictates the molecule's shape, how enzymes recognize it, and how our bodies metabolize it for energy. Finally, we learned how to mathematically represent these sugars using their empirical formula, and how to calculate their three-dimensional variations using the Lebel-Van't Hoff rule. You now have a solid understanding of the structural foundations of simple carbohydrates. Great job, and keep exploring the chemistry of life.